In this particular video, we are going to see the first classical experiment of this unit, that is Messelson and Stahl experiment. But before entering into the Messelson and Stahl experiment, we need to know about a basic concept called as three models of DNA replication. Already in our previous video, we have seen what is the central dogma of life or central dogma of molecular biology. And in the central dogma of molecular biology, the first step is replication. That is, a double-stranded DNA getting two copies of its own. So this DNA replication, how it's happening? It was like $1 million question in the minds of all the scientists. This came in after the discovery of the DNA structure by the Watson and Crick. And from the question that is raised in the mind of the scientists, there came three models. The models are the conservative replication, dispersive replication, and semiconservative replication. What is the difference in these three models? We will see. The first model is called as semiconservative replication. So in the semiconservative replication, there is going to be two strands of DNA which is going to unwind from each other, and each of these individual strand is going to act as the template for synthesis of a new complementary strand. This results in two DNA molecule with one original strand and one new strand. You could see the image here. So this is semi-conservative replication. So here, imagine this is the original strand, that is the parent strand. You could see the color here, it's completely blue in color. So now this parent strand, which is this blue in color, it's going to unwind from each other. That is, this double strand is going to unwind. So you get two individual strands, and these two individual strands are going to act as the template strand for the production of the new strand. So you get two DNA in the end, and in each of this DNA, one strand is going to be completely parent and the another strand is going to be completely newly formed. So this is the first model, that is semi-conservative replication. The second model is called as conservative replication. In this model, the DNA replication is result in one molecule that consists of both the original DNA strands, and another molecule is going to consist of two new strands. So I will share you the image. So this is your parent strand, which is blue in color. So this parent strand, when it undergoes replication as per the conservative replication model, it's going to produce two daughter DNA strands, and in which the first DNA molecule is going to have both the strands from the parent, and in the another DNA molecule, both the strands are going to be completely newly formed. This is the second model, that is conservative model. Now moving to the third model, which is called as dispersive replication model. In this dispersive replication model, this DNA replication is going to result in two DNA molecules that are mixtures or hybrids of both the parent and daughter DNA. In this model, each individual strand is a patchwork of both the original and the newly formed DNA. So this is the image of your dispersive replication. You could see here in both the strands of both the DNA molecules, you could see the blue as well as the yellow colors coming like with some intervals. So blue, yellow like that. So you could see the patchwork of both the parent strand and the newly formed strand in all the four strands of the two newly formed DNA molecules. This is the third model called as dispersive replication model. So now, after getting these three models, now scientists were like in huge confusion. Which is going to be the one that's really happening inside our body? Whether it's semi-conservative, whether it's dispersive, or it's going to be conservative. That's when the Messelson and Stahl experiment happened. Messelson and Stahl conducted their famous experiments on DNA replication using the E. coli bacteria as a model system. They began by growing this E. coli in a medium, or we call it as a nutrient broth, which contains a heavy isotope of nitrogen. Now you may get a question, why they specifically went for this nitrogen? They could have gone for any other elements, and why they specifically choose this nitrogen? Because if you see the structure of DNA, it's made up of the nitrogenous base pairs. That's why they specifically went for this nitrogen. And the two type of nitrogen isotopes that they opted for this particular experiments are N15 and N14. We'll see more about what happened in this Messelson and Stahl experiment in the next video.